In this video, we're going to focus our discussion on DNA replication and the molecular mechanism by which that occurs in cells. But before we do that, I just want to begin with a little bit of a refresher on the structure of DNA itself, because the structure of DNA and its biochemical makeup is centrally important to the mechanism by which it's copied. And as you all know, these two gentlemen that we see in the image here are James Watson and Francis Crick. They are famous for having discovered the structure of DNA, deoxyribonucleic acid, not for discovering DNA, but for discovering the structure of DNA. And what you have here is the title of their paper that they published in April of 1953. And the title was A Structure for Deoxyribonucleic Acid. And the beginning of that paper is just this very simple sentence, a bit humble. We wish to suggest a structure for the salt of deoxyribonucleic acid, DNA. This structure has novel features which are of considerable biological interest. And they could not have been more right, because in solving the structure of DNA, essentially they solved the puzzle of heredity and how genetic information can be passed on down the generations. So this was a seminal discovery in the history of biology. But it should be noted that they did not make this discovery in isolation. They were actually building on the work of a lot of people who had gone before, who had discovered some very important things about DNA that they were able to pull together finally in coming up with this structure, right? So the base pairing rules, for example, were not previously known, but what was known was that the amount of adenine that you found in a cell was roughly equivalent to the amount of thymine. The amount of cytosine was roughly equivalent to the amount of guanine. And Watson and Crick were able to take that information and pull it together and say, well, maybe these bases are actually pairing with one another in the structure of DNA, and that's why their ratios are always roughly equivalent to one another. Right? They were also building their structure based on X-ray crystallography results, which other people had derived, namely a couple of people that were working at King's College in London, Wilkins and Rosalind Franklin. Wilkins, who shared the Nobel Prize with Watson and Crick, and Rosalind Franklin, who unfortunately died before that Nobel Prize was awarded, and those prizes are not awarded posthumously. So anyway, they were building and using a lot of information and a lot of discoveries that other people had made, but really pulling it all together and solving the puzzle of the structure of DNA. Okay, so what was that structure that they had solved? Well, as we all know now, the structure of DNA, at least the most common version of DNA that we see in cells, what we call bDNA, is that it's a right-handed double helix made up of two nucleic acid strands that are hydrogen bonded with one another in anti-parallel orientation. And so that's sort of depicted here on the left. We've looked at this structure before. We know that there is a phosphodiester backbone, sort of illustrated by this ribbon structure that runs along the outside of the helix, with the nitrogenous bases of the nucleotides sort of tucked away to the interior, hydrogen bonding with one another, but also accessible from the outside via the major groove, where they're sort of exposed to the external environment. And we talked about the importance of that before in thinking about proteins interacting with DNA in order to change the way that DNA is being expressed, or whether it's being copied, or to regulate and control the function of DNA. And so this is sort of a schematic of that B version of DNA. So let's break this down and look at these terms that we're dealing with here to define the structure in a little bit more detail. So double helix is referring to the fact that there are two individual molecules of DNA that are hydrogen bonded with one another, right? Right-handed is referring to the direction of coil of the helix. So if you imagine wrapping your hand around this helix such that your thumb is pointing up, if the helix coils in a clockwise direction, in the same direction that your thumb is pointing, then by definition that's a right-handed helix. And that's exactly what we have here. If the helix was coiling up in an anti-clockwise or counterclockwise direction, in the direction of your thumb, then that would be a left-handed helix. So bDNA is a right-handed helix made up of two strands, hydrogen bonded. And this term anti-parallel orientation refers to the directionality of the phosphodiester backbone to which those bases are bonded. So we talked about this before, that the structure of an individual nucleotide is made up of a nitrogenous base that's bonded to a ribose or deoxyribose sugar, depending on whether you're looking at RNA or DNA. In this case, we have no hydroxyl group on this 2' carbon of the sugar, so this is a DNA nucleotide. A deoxyribonucleotide, and then bonded to the 5' carbon of this nucleotide, we have one or more phosphate groups, right? If it's already incorporated into a chain, it would be just one. So that's the structure of one nucleotide. And within each individual nucleic acid strand, these nucleotides are bonded to one another by their sugars and phosphates in a characteristic directional fashion, right? With the phosphate group of the next nucleotide in the chain being bonded to the 3' carbon position of the sugar of the preceding nucleotide. And so we have this sort of directionality where if we're looking down this phosphodiester backbone, we can sort of trace the linkages: phosphate attached to 5' carbon through the sugar, 3' carbon, phosphate attached to 5' carbon, 3' carbon, phosphate attached to 5' carbon, 3' carbon, phosphate attached to 5' carbon, and then here's our 3' hydroxyl group that is dangling off of the end. Now this is sort of implying that there's another nucleotide down here, but if there wasn't, then this would be a free hydroxyl group that's available for bonding by the next nucleotide to be added in the chain. So in terms of directionality, for this phosphodiester backbone, the directionality is 5' to 3' going top to bottom. If we look at this phosphodiester backbone over here, it's running 5' to 3' bottom to top, right? Here's our 5' phosphate, 3' carbon, 5' phosphate, 3' carbon. So the directionality here is going from the 5' end of this backbone to the 3' end. And again, down here we would have our free hydroxyl group available for bonding here. So our two strands are running in opposite direction with regard to their 5' to 3' orientation. Now, why is that? Well, it turns out that that's the only way that these phosphodiester backbones can pack close enough to one another to allow this hydrogen bonding interaction. So it's a structural steric limitation. Remember, these phosphate groups are huge. There's a lot of negative charge associated with them, and so there's both steric and a negative charge repulsion that's being dealt with here. And so the packing requires that they sort of be in opposite orientation. And then these bases are projecting off from the sugar and hydrogen bonding with one another. These are not covalent bonds. These are hydrogen bonding interactions between complementary functional groups on those nitrogenous bases in the nucleotides, right? So why does G hydrogen bond specifically with C? Well, they have matching complementarity. Why does T hydrogen bond with A? They have matching complementarity. And this is something that we looked at previously in the course, and we looked at this in some detail, so I'm not going to belabor it now. The consequence of this highly specific base pairing interaction is that if you know the sequence of one strand of DNA, then you automatically can predict the sequence of the opposing strand of DNA because you know the base pairing rules. If you were not able to see this strand on the right, you could predict that its sequence would be CAGT just by pairing up nucleotides with the left-hand strand, right? And in the cell, there are enzymes that can do precisely the same thing, that can pick up nucleotides based on correct base pairing interactions and incorporate them into a growing strand. And as soon as Watson and Crick had solved the structure of DNA, they immediately recognized this fact, and the fact that this highly specific pairing interaction would be used by the cell as a means of copying the DNA, right? And copying is what we're talking about when we're talking about heredity of two daughter cells produced by cell division inheriting the genetic instruction manual from the parent cell. That's heredity, right? We can think of it sort of on the macro scale in terms of organisms reproducing and producing other organisms, but that's all really at the cellular level, passing on your DNA. So how does a cell pass on its DNA in high fidelity with each round of cell division, well, it's simple enough. You just separate the two parent strands of DNA, use each as a template to generate a new daughter strand opposite by the base pairing rules, and in that manner generate two DNA helices, which will each become the instruction manual for the two daughter cells. And Watson and Crick were absolutely aware of the seminal importance of this discovery, and towards the end of their very short one-page paper, they actually have this sentence that's alluding to that fact. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. And of course they were right. We now know the mechanism by which DNA is copied, and it's exactly what they predicted, that the two parent strands would be separated and each would serve as the template for a new opposing complementary strand by the base pairing rules. So this mechanism of DNA replication is referred to as semi-conservative DNA replication, because in each of the new DNA double helices you have one old parent strand and one newly synthesized daughter strand. So in other words, the parent strand is semi-conserved in each of the new daughter strands. Right? So what we have is a situation where a template strand that consists of old parental DNA, so the two strands of the parent DNA would have separated, and here we're looking at just one of those strands. In this case, the strand that runs from 5' to 3' bottom to top would be serving as a template for the enzymes, which are called polymerases, that will copy this and produce the new opposing strand that's going to be hydrogen bonded to it. And it's going to do that by the base pairing rules. So essentially following along this template strand and adding in bases that match the sequence in that template strand by the base pairing rules. So opposite this T, there will be an A, opposite this G, there will be a C, opposite this C, there will be a G, and so on, right? So you have to imagine there's a whole pool of nucleotides out here in the cell, and the enzyme that's copying DNA is really selecting from that entire pool and picking up the nucleotide that matches or sort of slots into position because it has the correct hydrogen bonding interaction with the nucleotide in the chain. And as we talked about before, the way that this incoming nucleotide is going to be added on to this growing chain is by a new covalent bond that's going to be formed between this first phosphate on this incoming nucleotide and the 3' carbon of the sugar of the preceding nucleotide. So essentially this oxygen is going to replace one of the oxygens of the phosphate group itself. This covalent bond between these two terminal phosphates is going to be cleaved, and this phosphate is now going to be joined here, right? So it's now going to be incorporated into this position in the chain, and those two terminal phosphates are going to leave as a pyrophosphate group. So again, this set of reactions is catalyzed by a set of enzymes collectively called DNA polymerases, and the enzymes that would be copying DNA to produce RNA would be catalyzing a very similar mechanism, except that they would be recognizing RNA nucleotides, which would have that additional hydroxyl group here on the 2' carbon, as we talked about before. So those would be RNA polymerases. So DNA polymerase is the class of enzyme. There are many different specific subtypes that have specific roles to play, as we'll see, that copy DNA to produce more DNA. RNA polymerases copy DNA to produce RNA. So DNA polymerases are involved with DNA replication, and RNA polymerases are involved with transcription. So make sure that you keep those two classes of enzymes distinct from one another in your mind, even though they're catalyzing essentially the same mechanism. All right, so one thing that you need to understand is that this mechanism that we've just talked about, where the incoming nucleotide is going to be added on to the 3' carbon of the pre-existing chain, is the way that all known DNA and RNA polymerases work. So in other words, all known polymerases have evolved to only synthesize nucleic acids in the 5' to 3' direction. If we think about this strand that's growing here, it's growing in this direction, right? From the 5' end and getting longer on the 3' end, right? And that's the way that all polymerases work, which imposes some limitations and causes some difficulties when we start thinking about copying DNA, because of course DNA is double-stranded, and those two strands of parental DNA are going to be running in opposing directions. So that introduces a little bit of a problem that the cell has to deal with, and we'll talk more about that in just a moment. So an important thing to recognize about DNA replication is that it's going to begin at specific sequences, specific locuses or loci in the genome, no matter what organism you are talking about. And those specific sequences of DNA where the copying of DNA begins are referred to as origins of replication. And if we look at a simple prokaryotic cell like an E. coli cell, what we would find is that the entire genome of that organism is maintained in one circular chromosome. Now, there are those extra chromosomal elements, the plasmids that we talked about before, that have non-essential genes. But the main set of genetic instructions is in that one circular DNA molecule, and it consists of approximately 4.6 times 10 to the 6 base pairs. So about 4.5 million base pairs is the entire genome of an E. coli cell. That circular chromosome is well served by one origin of replication. So if we were looking at replication in E. coli, what we would see is this circular chromosome being replicated beginning at this origin, which in E. coli is referred to as ORI-C, and we would see separation of the two parental strands, illustrated in dark blue here, and that's going to form what's referred to as a replication bubble. And we would see synthesis of two new daughter strands here in light blue that are complementary to these two parent strands. And that replication would proceed in both directions away from that origin, with both strands of the DNA being copied at the same time, so that you can see this dark parental strand is being paired now with a newly synthesized daughter strand, and this dark parental strand is also being paired with a newly synthesized daughter strand. And same thing over here on the other side of the replication bubble. So in other words, replication is bidirectional from those origins. And that's true for any organism that you would be dealing with. There are a few exceptions to that. There are some viruses that don't have bidirectional replication. There are some plasmids that don't have bidirectional replication. But for the most part, the rule is the replication begins at a specific origin and proceeds bidirectionally, with both strands being copied from that origin, right? And so in an E. coli cell, what you would see is this replication bubble getting bigger and bigger and bigger as the DNA is separated here at what's referred to as a replication fork. This is the point where the parental DNA strands are being peeled apart. So you have two replication forks for each replication bubble, and synthesis would be going all the way around this chromosome in this direction, all the way around the chromosome in this direction, and eventually that bubble is going to expand and get to the other side of the DNA, and you would have two DNA molecules, two chromosomes, sort of intertwined, and they would be enzymatically separated to yield two completely separate daughter DNAs that would end up in the two daughter cells produced in cell division. All right, so that's the situation that you'd see in a prokaryotic cell in general. We just happen to be looking at E. coli here. Now, if you move to the eukaryotic world, we know that eukaryotic cells in general do not have circular chromosomes. They have multiple linear chromosomes. And so if we look at a yeast cell like S. cerevisiae, Baker's yeast, what we would see is that the genetic content of that cell is encompassed by 16 linear chromosomes and 12 times 10 to the 6 base pairs, so 12 million base pairs of DNA. And those linear chromosomes would have 400 replication origins, right? So more than one replication origin per linear chromosome. And if we look in human cells, we would see something even more dramatic: 46 linear chromosomes, 3 times 10 to the 9th base pairs of DNA being served by 40,000 replication origins scattered along those linear chromosomes. Well, not scattered. That implies that it's random. It's not random. It's specific sequences, but sort of distributed along those 46 linear chromosomes. And so if we looked at the linear chromosomes in a eukaryotic cell, what we would see is multiple origins of replication along that double-stranded DNA. Each of those origins would be separated, opened up to form a replication bubble. We'd have 2 replication forks at each of those replication bubbles moving in opposite directions, with both strands of parental DNA being copied at the same time and newly synthesized strands being created opposite them. And so you'd see this replication bubble expanding out in both directions, and likewise for all of the replication bubbles along that linear chromosome. And they would keep expanding until eventually they'd run into one another, and again we would have 2 completely distinct new DNA daughter molecules, each of which is composed of an old parental strand and a newly synthesized daughter strand, right? And so then these would be separated from one another and sent to the 2 daughter cells that are produced during cell division. All right, so now we understand in general overview what happens in DNA replication. What we want to do now is delve into the details. How is this DNA being copied? What are the enzymes and other proteins that are involved, that are required in order to make this happen? And what are the enzymatic activities that are involved here? So we're going to delve deeply into the details here. And what I'll tell you is that the mechanism by which DNA replication occurs in a prokaryotic cell like E. coli and a eukaryotic cell like a yeast cell or a human cell, the mechanism is virtually identical, right? But what's different is the complexity of the process. And so what happens in E. coli involves a lot fewer specific molecules and enzymes. And what happens in eukaryotic cells, basically you take the number of proteins and enzymes in E. coli and triple it or quadruple it. And so we're going to be looking at DNA replication as it happens in E. coli because it's a more simple model of the process, but everything that we learn about can be applied to eukaryotic cells. It's just that the complexity and the number of proteins and enzymes and other players would be dramatically increased. So we're going to be focusing on E. coli DNA replication as a model of what happens in all living organisms. So we're going to zoom in to a replication origin, and even more specifically to one replication fork that's proceeding from that origin in one direction. So just to make sure that everybody's understanding what we're looking at here. Wow, that was really a thick line. We're talking about a replication bubble. This would be a replication fork where the 2 parental strands of DNA are being separated from one another, and this fork is opening up the parental DNA in that direction. There'd be another fork over here that we're not looking at right now. Okay, so we're sort of zoomed in looking at this replication fork, and these are the 2 parental strands that are being separated. All right, so at the initiation of DNA replication, there are other proteins that recognize the origin, that open up the DNA, and sort of set the scene for DNA replication to begin. We're not going to focus on those guys. We're going to talk about what's actually happening at this replication fork to copy the DNA. So first thing you need is an enzyme that's going to be peeling these 2 parent strands apart. And that is our helicase enzyme that's illustrated here as this green blobby protein. So helicase is responsible for unwinding the parental DNA strands. So even though DNA polymerase is going to be copying the DNA, it can't separate the 2 parent strands. It needs to have that done for it. And so helicase is responsible for doing that. Now remember that what is holding these 2 strands together is hydrogen bonding interactions between base pairs, right? So what would happen if this helicase came through and separated the 2 strands and then just moved on down the line? Well, those 2 parental strands would quickly re-find those hydrogen bonding interactions and go back together. Right? And so to prevent that from happening, we have a set of proteins called single-strand binding proteins that prevent the DNA strands, the single strands of DNA, which we'll just say ssDNA, so prevent single-stranded DNA from re-annealing. And by re-annealing, we mean reforming a double helix, right? So finding those hydrogen bonds. So single-strand binding proteins would come along and bind to both of the separated parental strands and prevent them from finding one another again and forming hydrogen bonding interactions. Now, as you can imagine, this change in the structure of DNA, peeling apart the 2 strands of DNA, causes a dramatic strain on the DNA helix ahead of that stress being applied. And so you'll actually see supercoiling being introduced and even a potential for tangling to happen as these two parental strand coils are being peeled apart. And so there's a whole set of enzymes called collectively topoisomerases that are charged with maintaining the structure of DNA and relieving that stress and that supercoiling ahead of the replication fork. So topoisomerases in general untangle and remove supercoils so that the helicase can get in there and separate those two strands, right? And there's a whole set of proteins that do that. Okay, so we've separated the two parental strands. We've dealt with some of the stress and torsion supercoiling that's going to happen ahead of that. We've prevented the two strands from reannealing. Now we're actually ready to see enzymes come in and start reading the DNA sequence and laying down a new strand opposite that parental strand. Now, it's a funny fact about DNA polymerases that there's no identified DNA polymerase so far that we know of that can actually sit down on a single strand of DNA and just start copying, right? There's no DNA polymerase that can come along, sit down right here and say, oh, okay, there's an A, I'm going to put a T here, there's a G, I'm going to put a C here, right? There's no DNA polymerase that can do that, what's referred to as de novo synthesis. DNA polymerases in general cannot sit down and start a new strand of DNA. All they can do is add on to an existing strand of DNA. And so they have to have some sort of what's referred to as a primer to get them going. And so in DNA replication specifically, what we see is that there's an enzyme that starts the process off called primase or RNA primase. And primase is responsible for synthesizing a short segment of RNA, ribonucleic acid, opposite the single-stranded parental DNA. So it's going to synthesize a short 10 to 20 nucleotides long RNA primer to begin the process. And once that primer has been laid down, now you can have a DNA polymerase, which I'll just abbreviate as DNA Pol, come and bind to the end of that primer and start adding nucleotides on in the 5' to 3' direction. So, but it needs a primer to add on to. It can't just sit down and start synthesizing. So I mentioned before that there are multiple DNA polymerases, and that's true even in a simple cell like E. coli. So there are two specific DNA polymerases that are involved with DNA replication, specifically in E. coli. There are a whole bunch of other DNA polymerases that are all having different specific roles to play in the life of the cell, for example, in DNA repair, right? But the two that we're going to be looking at are the ones that are involved with DNA replication specifically. And those are DNA polymerase 3 and DNA polymerase 1. And we're going to look at each of those individually. So DNA polymerase 3 in E. coli is what's referred to as the main replicative polymerase. It's what's responsible for doing the vast majority of the DNA synthesis during DNA replication. All right? So mechanistically, what we're talking about here is the DNA polymerase that does what we just referred to, right? Comes and binds to the end of the RNA primer and adds on DNA nucleotides, extending that primer as it goes. So this would be specifically DNA polymerase 3 adding on to that RNA primer. DNA polymerase 3 is actually a very large complex of multiple polypeptides. More than 15 individual protein or polypeptide subunits come together to form this overall enzyme that we call DNA polymerase 3. Now, you're not going to have to learn what the vast majority of these do. There are only 4 that you're going to need to know the functions of, and those are the alpha subunit, the beta subunit, the epsilon subunit, and the tau subunit. All right, so these individual protein subunits all have specific functions and or enzymatic activities that are associated with them during the overall process of DNA replication. So let's see what they're actually charged with. The alpha subunit is what is responsible for actually synthesizing the DNA. And so the alpha subunit is an enzyme. This is a polymerase. It has 5' to 3' polymerase activity. And so it's going to be reading the parental template strand and synthesizing a new strand opposite that template using the base pairing rules in the 5' to 3' direction. That's what we mean by having 5' to 3' polymerase activity. The epsilon subunit has a very different enzymatic activity. It's actually a 3' to 5' exonuclease. Now, we've talked about nucleases before in the class. Nucleases are enzymes that degrade nucleic acids. An exonuclease would be an enzyme that degrades a nucleic acid from the end, right? So essentially binding to one end of the strand or the other and chewing away individual nucleotides. So you can sort of imagine that as being an enzyme that comes in here from the end and chews away individual nucleotides. Now, in this case, this is a 3' to 5' exonuclease, so it specifically recognizes a 3' end and chews away in that direction, right? There are other nucleases that would bind to the 5' end and chew away in that direction. So that's what this directionality is referring to. And that's not going to make a whole lot of sense right now, but we're going to come back to that and explain that exonuclease activity in just a moment. The beta subunit is what's referred to as a clamp. It is a subunit that has a sort of a donut shape with a hole in the middle, and it surrounds and clamps this whole complex onto the DNA that is being copied. And so in doing so, it actually increases the efficiency of the polymerase complex of copying DNA. If that beta subunit clamp did not hold the rest of the complex onto DNA, then every 20 to 50 nucleotides, this whole complex would fall off and copying would come to a halt and it would have to be reloaded back on. So the beta subunit prevents that from happening. It clamps the polymerase complex, the rest of the polymerase complex, onto the DNA and thereby increases efficiency and what's referred to as processivity. All right, so what's the function of the last subunit? The tau subunit, you can think of T stands for tether, and what we have to imagine is that we've got two parental strands of DNA sort of coming along like this, pardon my terrible helix, and one of those parental strands is going to be copied by this polymerase complex on the left, and the other strand is going to be copied by this polymerase complex on the right. Remember, we said DNA replication is bidirectional. So with both parental strands being copied at the same time. So at each replication fork, you're going to actually have at each replication fork, you're going to have a polymerase complex binding to each of the parental strands and synthesizing a new daughter strand opposite, right? The tau subunits and the rest of the subunits that we're not going to go into their functions tether these two complexes on the two parental strands to one another so that they don't essentially run away from one another, right? So it's sort of coordinating the bidirectional and double-stranded replication of DNA so that it's happening in an organized and coherent fashion. And just to make sure that that was all clear, I've also given you that sort of typed out here, all of those functions associated with those individual subunits. So you have that. What about DNA polymerase I? Well, the role of DNA polymerase I in DNA replication is very different. As you can imagine, we don't want RNA in our DNA. We want our DNA to be consisting of only DNA, nucleic acid, hydrogen bonded with one another to form our nice cohesive DNA double helix. So all of those RNA primers that get laid down to start off the new strand of DNA eventually have to be removed and replaced with DNA. And that's what DNA polymerase I does. It replaces the RNA primer with DNA nucleotides. Now, the structure of DNA polymerase I is considerably more simple than DNA polymerase III. It's a single polypeptide, one protein. But interestingly, that one protein actually has three different enzyme activities. It has three different active sites that can catalyze three very different reactions. Like DNA polymerase III, it's going to have a 5' to 3' polymerase active site, so it's going to be adding nucleotides onto a chain, synthesizing DNA using the parental DNA strand as a template. Like DNA polymerase III, it's going to have a 3' to 5' exonuclease activity, which, as we're going to see, is very important for proofreading. So it has that same exonuclease activity that DNA polymerase III does. It also has a 5' to 3' exonuclease activity that is not used during DNA replication, but is very important for DNA repair, because it turns out that DNA polymerase I is a critical enzyme that participates in a lot of the DNA repair pathways that are going on in E. coli, but it's not used for DNA replication. And I have to say that your textbook still contains an error, as it has from previous editions, that says that DNA polymerase I removes the RNA primer with this exonuclease activity, and it does not. It just replaces that primer with DNA. There's another enzyme that actually removes the primer, but we'll come back to that in just a minute. All right, so let's talk a little bit more about this exonuclease activity and its importance during DNA replication. Why do we need to have an exonuclease? Well, it turns out that the error rate of the polymerase activity of DNA polymerase is rather high. It's unacceptably high. We'd be introducing thousands of new mutations in every round of replication based on how often polymerase makes a mistake. And so misincorporation of nucleotides during DNA replication happens. So this is illustrating a polymerase active site, all right? You can sort of think of it as a hand sort of wrapping around the DNA that's being copied. And in this case, this is our parental strand here that's running 3' to 5' in this direction. And this is our newly synthesized strand in the lighter blue. So here's our active site that has the polymerase activity. It's reading the sequence of nucleotides in the parental strand and synthesizing a partner, right, opposing that by the complementary base pair rule. So if this is a T, it's going to be adding in an A. If this is a C, it's going to be adding in a G as it synthesizes and extends this 3' end of the growing strand. But what if this is an A and this polymerase activity accidentally adds in a C, makes a mistake and adds in a C? That's a mismatch. And essentially this enzyme is going to be able to feel that that pair doesn't feel right. It doesn't feel like a correct base pairing interactions, because these two are not going to match up the way that they should if they had true complementarity in their hydrogen bond donors and acceptors. And so what will happen then when a mismatch is detected is that this end of the newly synthesized DNA is going to get flipped down to the other active site, which is the exonuclease activity. And this exonuclease activity, again, we can imagine as a sort of Pac-Man, is going to chew away the nucleotides that were just incorporated. And how many get chewed away is sort of random. It's going to be on the order of 5 to 10, maybe even 15 nucleotides. So essentially, it's like a delete button, right? Delete, 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 delete. It goes back. And then the end gets flipped back up to the polymerase, and this enzyme gets a do-over. So mismatch occurs. That strange shape associated with incorrect base pairing is detected by the enzyme. That stimulates a shape change that flips that end down here to the exonuclease active site. Exonuclease active site chews away the nucleotides that were just incorporated and is transferred back up to the polymerase active site, and the enzyme picks up where it left off and hopefully gets it right this time and doesn't misincorporate nucleotides. Now, it seems counterintuitive that you'd have an enzyme activity that could make such mistakes, right? It seems like it should be better at being able to feel out the hydrogen bond donors and acceptors and figure out whether they are matching up correctly. And you're right, it is counterintuitive. However, what we haven't talked about is that the nucleotides themselves change their structure. And so effectively, they can fool the polymerase activity into thinking they're a different nucleotide. And so to understand that, we have to look back at the structures of the nucleotides and understand something called tautomerization. So here's the structure of the thymine nitrogenous base as it would most commonly be observed in a cell, right? So biochemically, this is the most stable structure that we should see for the thymine base. However, at some low frequency, what we can see is something called a proton shift, where a functional group, in this case an amino group, loses a hydrogen that takes up residence on another functional group, in this case a carbonyl, changing the functional groups and the patterns of hydrogen bond donors and acceptors. If you think back to our hydrogen bonding discussion way back at the beginning of the course, we talked about the difference between hydrogen bond donors and acceptors, right? We talked about the fact that a hydrogen bond donor is the functional group that donates the partially positive hydrogen to the hydrogen bonding interaction, right? So in a normal thymine-adenine base pairing interaction, this amino group is going to be the hydrogen bond donor, and this ring nitrogen on adenine is going to be the hydrogen bond acceptor. Right? This is a partially positive hydrogen. This is a partially negative nitrogen because it's bonded to carbon on either side. And we can see an attraction between those partial positive and partial negative charges. Likewise, this carbonyl oxygen is our partially negative hydrogen bond acceptor, and this amino hydrogen is our partially positive donor. And so we have complementarity in our donors and acceptors. If we get this proton shift where now that proton has moved to this oxygen, well, now this no longer can be a donor, right? And this becomes a donor or an acceptor. Basically, the hydrogen bonding pattern gets disrupted. Well, it turns out that when you have this kind of a proton shift, you can get anomalous base pairing interactions where now you have new complementarity that's been introduced, right? So all four of the nitrogenous bases can undergo these nitrogenous shifts, which change them from their more common keto and amino forms to their minor enol and imino forms. And depending on which specific base you're looking at, you're going to get different specific newly introduced hydrogen bond donors and acceptors, right? So this is an equilibrium. And the nitrogenous bases are constantly shifting back and forth. Most of them are going to be found in the common form, but at brief instantaneous flashes of time, they're going to be present in this rare form before they shift back. So really this should be a very small arrow in that direction and a very large arrow in that direction. This is much more common in the cell, and that's true for all of these nucleotide bases. All right, so what can happen when you have a base that has shifted to its rare, less seen tautomer is that you can have non-Watson-Crick base pairs, anomalous base pairing arrangements, where you could have a normal adenine now matching up by those hydrogen bond donor and acceptor interactions with cytosine when it's in its rare form, right? Its rare amino form. So when that happens, if cytosine happens to be picked up by DNA polymerase, it's going to think this is a thymine based on hydrogen bond donors and acceptors, right? And so it could incorporate or misincorporate a cytosine at a position where it should be picking up and adding in a thymine, opposite an adenine in the template strand. Likewise, you could have a thymine in the template strand and the DNA polymerase just so happened to pick up a guanine that has shifted to its rare tautomer form, and now we're going to see complementary base pairing interactions between thymine and guanine. And so we have to think of this like puzzle pieces. There aren't any letters on these nitrogenous bases in a cell. All polymerase can do is feel out whether these two bases are matching one another and their hydrogen bond interactions are bringing them close enough to one another that the structure seems to be correct. And so these nitrogenous bases in their rare tautomer forms can trick or fool the polymerase into misincorporating nucleotides into the DNA during synthesis. And when that happens, you have the possibility, the potential of introducing in the DNA mutations, right? This is how one way that mutations can be introduced during DNA replication. So let's say that we have our parental DNA. These are our two strands of the double helix being separated through the process that we just talked about. So this is DNA replication. This would be a DNA replication fork. And we have our polymerases reading these parental strands and synthesizing new daughter strands opposite. So let's say this strand stays in its normal tautomeric form. All of these nitrogenous bases stay in their normal, more common forms. Polymerase is going to come along and it's going to copy this single strand of DNA, and it's going to correctly synthesize the opposing strand by the base pairing rules, no problem here. But let's say that this adenine shifts to its tautomeric structure such that it now will hydrogen bond to cytosine, and polymerase comes along and picks up a cytosine and incorporates that opposite that adenine, right? So what we get here is a mismatch, an AC base pair instead of a normal AT base pair. Now this adenine, we have to say, is going to shift back to its more common form, right, eventually. And so now we're going to have a disruption in this base pairing arrangement where they're no longer going to match one another. And that's going to happen fairly quickly because, as I said, the common forms are more favored structurally, and the rare tautomers are just present for a brief instant in time. So after this misincorporation, there's a window of time where the cell is going to be able to recognize this as a mismatch. The cell has enzymes that are constantly surveilling the DNA and looking for weird distortions that are caused by incorrect base pairing, these sorts of mismatches, and will recruit DNA repair enzymes to remove that incorrect, newly incorporated bit of DNA and replace it with the correct sequence, right? So this can be a stimulus for mismatch repair to occur, but if the cell doesn't detect this mismatch and repair it before the next round of DNA replication, presumably that adenine is going to shift back to its normal base pair, right? And so one of the DNA daughters that's produced is going to be normal, but this C that was misincorporated in the previous round of replication, the cell has no way of knowing that C is not supposed to be there. And so when these two strands get separated from one another, that's going to specify a G opposite. And this is no longer a mismatch, right? This is a normal CG base pair. So the cell can no longer recognize that a mutation has been introduced. And this will be a long-standing mutation that persists in the genome then. And this is one source, a major source of those single nucleotide polymorphisms that we talked about in the errors of meiosis discussion, right? So this is now a single nucleotide base pair change. So the daughter cell that inherits this DNA now has a mutation at this position that will exist in the genome going forward. Okay, so now that we've looked at the molecular players and we know the enzymes and other proteins that are involved in getting us going, let's zoom back out and look at the overall process once again and return to the problem that I alluded to earlier, which is the anti-parallel orientation of the two strands of parental DNA and the fact that DNA and RNA can only be synthesized in the 5' to 3' direction, because that introduces a problem, right? If we think about it. If we imagine an origin of replication with, again, our two parental strands of DNA, here illustrated as the dark blue, and we add some directionality onto this. So let's say this is the 5' end, and this is the 3' end. We know that because of the anti-parallel nature of our DNA double helix, that this must be the 3' end of this strand, this must be the 5' end of this strand, right? And so if we're thinking about directionality, this parental strand is running 5' to 3' left to right. The newly synthesized daughter DNA that is being created opposite that by the complementary base pairing rules is going to have an anti-parallel orientation. It's going to have to be 5' to 3' right to left, right? And that is the direction that DNA is synthesized. And so if we think about it, this DNA molecule that's being synthesized here opposite that strand of DNA is going to be running 5' to 3' in this direction, right? But on the opposite strand here, which is 3' to 5' left to right, the newly synthesized daughter strands are going to have anti-parallel orientation going the other way, right? So 5' to 3' goes left to right for these guys. So this is a problem if we're thinking about copying these two parental strands of DNA, because what this says is that we've got a replication fork that's opening up in this direction. We've got a helicase protein that's unwinding the parent strands of DNA in this direction. And so we're trying to copy, we have an overall direction of synthesis that's going right to left, but only one of these newly synthesized daughter DNA molecules can be synthesized right to left. These DNA molecules have to be synthesized left to right because that's 5' to 3' for them. And so these newly synthesized daughter strands that have to be synthesized in the opposite direction to the direction that the helicase is opening up the double helix have to be synthesized in what's referred to as a discontinuous fashion. So this is discontinuous synthesis, and they have to be synthesized as short little fragments of DNA, what are called Okazaki fragments, whereas the other strand of DNA that's being synthesized in the same direction that the helicase is opening up the replication fork can be synthesized continuously. As that helicase moves along and opens up the DNA double helix, the DNA polymerase just continues to synthesize, moving along right behind that helicase enzyme. Right? So we have one strand that's synthesized continuously, and that's called the leading strand. And then we have the strands that are synthesized discontinuously, and those are referred to as the lagging strand. Right? Now, the whole big picture idea here is the reason we have a leading strand and a lagging strand is because all DNA and RNA has to be synthesized. The only way those enzymes work is that they can only add on to DNA on the 3' end. So all DNA synthesis goes 5' to 3'. The problem that's introduced by that is that 5' to 3' goes in two different directions for the two strands that are being synthesized, right? One of those is going to match the direction that the helicase is moving, and the other is not. And so we're going to have some way to deal with that, and I'm going to show you an animation that makes clear how the replication machinery actually deals with this discontinuous synthesis, because it's pretty amazing and elegant. Now, the thing to recognize is that over here on the other replication fork coming from this same replication bubble, we've got another helicase enzyme that's moving in the opposite direction. And so we have an overall direction of replication for this fork that's moving in this direction. All right, so now let's think about our two strands of DNA and which strand is going to be synthesized in the same direction the helicase is moving. In this case, what we're looking for is 5' to 3' synthesis, right? So that's this bottom strand, right? The orientation of this parental strand is 5' to 3', right to left, which means that the other strand has to be 5' to 3', left to right. And so for this fork, the bottom strand is the leading strand, and the top strand is the one that's going to have to be copied discontinuously in order to synthesize these new DNA molecules 5 to 3, right to left. Right, so at each origin of replication, at each replication bubble, you're going to have two replication forks moving in opposite directions, with both parental strands being copied at each of those replication forks, but the newly synthesized daughter DNA is going to consist of a continuously synthesized leading strand at each of those forks and discontinuously synthesized lagging strands at each of those forks. All right? So here's another view of that same replication bubble with the two replication forks. Again, we're just zoomed in on this replication fork on the left-hand side. So in either case, it's important to recognize that there's going to have to be an RNA primer laid down to begin that synthesis, right? So these little red bits here are illustrating the RNA primer that was laid down by primase. So don't confuse yourself by thinking that the leading strand didn't have an RNA primer. It did. It was just that it only required one RNA primer right at the beginning, and then the polymerase, DNA polymerase 3 was able to extend that with DNA going forward in a continuous fashion. Whereas each of these lagging strands is going to be having its own RNA primer. All right, so if we zoom back in and look at this with some of the enzymes and other protein players here, we get something that looks like this. So here's our DNA polymerase III, which you'll recall is the main replicative polymerase that's responsible for extending the RNA primer with DNA, right? So here's our DNA polymerase III extending that DNA continuously in the same direction that our helicase is moving. Our overall direction of replication is right to left in this case for this fork. Here's our helicase. And then on the lagging strand, what's going on is the same thing that happened up here. There's a primase that lays down a primer. Once that primer is established, DNA polymerase III binds to the end of the primer and extends it with DNA. So the function's the same, right? And so you have the synthesis of a fragment of DNA here, and as the helicase opens up this replication fork and more single-stranded DNA here is now exposed, primase is going to come along, lay down another primer, then DNA polymerase III is going to come bind here, right, and extend that primer with DNA. And so that happens processively. As this helicase opens up the replication fork more and more, we'll get another primer laid down, another short fragment of DNA established, and so on as the replication fork moves in that direction. All right, so where does DNA polymerase I come in? Well, DNA polymerase I, recall, is the enzyme that's responsible for replacing the RNA primer with DNA. Now, as I said, it's not the enzyme that's responsible for removing the RNA primer. That's actually a separate enzyme called RNase H. It's a nuclease that specifically comes in and chews away the RNA primer, leaving a gap here. And when that gap is introduced, then DNA polymerase I is going to bind to the end of the DNA and extend that DNA and fill in that gap left by the primer with DNA, right? And so that happened up here, that's going to happen here. Everywhere there was an RNA primer, RNase H chews away that primer, leaving a gap in the sequence that DNA polymerase I will then come bind to and fill in, but again, synthesizing in the 5' to 3' direction. All DNA and RNA polymerases synthesize DNA in the 5' to 3' direction. Okay, so if we think of this, what overall is going on in the process of DNA replication, whether we're talking about the leading strand or the lagging strand, it's going to be the same. Single-stranded DNA is exposed. RNA primase synthesizes a short little stretch of RNA primer. DNA polymerase III is going to bind to the end of that primer and extend it with DNA. An enzyme like RNase H is going to come along, bind to the primer, and chew it away with its exonuclease activity. DNA polymerase I will then replace that missing sequence that was the RNA primer with DNA. And then we have an enzyme, DNA ligase, that comes in and causes the formation of a new covalent bond between those fragments of DNA that were produced. Now, I should note that these discontinuously synthesized fragments of DNA are referred to as Okazaki fragments, and those are named after Reiji Okazaki, who's the person who first provided the experimental evidence for this discontinuous synthesis to solve the problem of anti-parallel orientation and how the cell deals with that during DNA replication. So these discontinuously synthesized DNA molecules we refer to as Okazaki fragments. And DNA ligase is responsible for sealing the nicks in the phosphodiester backbone between those Okazaki fragments once the RNA has been replaced with DNA. Now, in E. coli, this mechanism by which the primer is replaced by DNA polymerase I by binding to the upstream bit of DNA and extending to fill in that gap works just fine, because remember, we're talking about a circular chromosome here. We start at an origin of replication. The two replication forks proceed all the way around that circular chromosome until they meet at the other side. But this is one continuous DNA molecule. When we think about the mechanism by which the primer is replaced, that mechanism is essentially the same in eukaryotic cells. As I said, all of this process is basically mechanistically the same. It's just that we have a lot more players involved, but the mechanism is identical. There's going to be a polymerase that has to bind to a bit of upstream DNA and fill in the gap that's generated when the RNA primer is removed enzymatically, right? So there are no gaps in our DNA double helix. In eukaryotes, this becomes a big problem, though, because eukaryotic cells have linear chromosomes, right? So the fact that we have linear chromosomes introduces what's referred to as the end replication problem. And the problem is that we cannot effectively replicate the 3' ends of our linear chromosomes. And so this is represented sort of schematically here. Now we're looking at a replication fork that's moving so that the direction of DNA synthesis overall is left to right here. And in this case, our top strand of parental DNA is 3' to 5' in the left to right direction. So if we're just focusing on this replication fork, this would be the 3' end of this parental DNA molecule. This would be the 5' end of that parental DNA molecule. And so this newly synthesized daughter strand is being synthesized 5' to 3' as we talked about before. So this would be our leading strand, right? And then at this replication fork, these would be our lagging strands. Now, the coloring is opposite to what we were just looking at, so don't confuse yourself. In this case, in this diagram, the RNA primer is actually in blue. So these little bits here are our RNA primers that have not yet been removed. These are the DNA bits that were added on to the RNA primer by the eukaryotic equivalent of DNA polymerase III. So the RNA primers were extended with DNA, and you can see that there's a gap here in the phosphodiester backbone where this little bit of DNA is not joined to the RNA ahead of it, right? So there's a little gap. So we're going to see these gaps in our sequences that, as we said before, are going to be sealed with a new covalent bond by our DNA ligase enzyme. But ignoring that for right now, let's think about what has to happen when this bit of RNA gets chewed away enzymatically, and we now have a gap here in this sequence. All right, so let's jump down to a little bit further along in the replication process. So let's say that that's already been effectively accomplished, and now we're further along here. So we have another bit of RNA here, primer, another bit of RNA here, and this bit of RNA had DNA added onto it. We've got a little gap here. So when this RNA is removed and this RNA is removed and we're left with a gap, the eukaryotic equivalent of our DNA polymerase I is going to bind to the end of this DNA that's upstream. So this would be the 3' end of the DNA, and it's going to add onto it with DNA, right? Extending it until it runs into this bit of DNA ahead of it. And we're still going to have a gap, but the gap is moved, right? Same thing's going to happen here. So the eukaryotic equivalent of DNA polymerase I is going to bind to this 3' end of the DNA fragment, extend it with DNA to replace that sequence that was removed, the RNA primer that was removed, and we're going to have a gap. Just like in prokaryotes, those gaps are going to be sealed. But look what happens here at the 3' end. There's no DNA up here that the eukaryotic equivalent of DNA polymerase I can bind to in order to extend it to fill in this missing sequence. So with every round of DNA replication, the very 3' ends of linear chromosomes cannot be effectively copied. And so the newly produced daughter DNA molecule is going to have some missing sequence. It could be hundreds to thousands of nucleotides long, depending on how these fragments get laid down. Now, why is this a problem? Well, what is DNA? DNA is your genetic instruction manual, right? It's your blueprint for life. These are genetic sequences that encode protein products, usually, that the cell requires in order to do all of the work of a cell. And so if we are basically cutting short our replicated DNA sequence with every round of DNA replication, then our DNA is getting shorter and shorter and shorter with every round of replication. And eventually what's going to happen is that we're actually going to start losing genetic sequence and genes. And that's a problem. So how do eukaryotes deal with this problem? Well, there's no way to stop this from happening. And so what happens instead is that in eukaryotic cells, there's an enzyme that early in embryonic development and in some special populations of cells throughout the adult life of the organism actually extends this 3' end of DNA with repetitive DNA sequence. So effectively adding sort of nonsense DNA, it doesn't encode anything, to the 3' ends of our chromosomes, so that during every round of replication, when we can't effectively copy this DNA, it doesn't matter because it's nonsense DNA that doesn't actually encode anything. And so these extensions of the 3' ends of DNA are referred to as telomeres. And they are added by an enzyme called telomerase. And so this doesn't stop the end replication problem from happening. It just makes it so that the end replication problem doesn't matter because we're not actually losing real genetic sequence here. So how does telomerase do this magic? Let's take a look at the mechanism here. So this requires 2 molecular components. Right? So there are 2 molecular components involved here. There is an enzymatic subunit that's associated with our telomerase activity. So this is the telomerase enzyme itself, the big blue blobby thing. And it turns out that this telomerase enzyme carries with it its own template in the form of an RNA molecule that allows it to bind to the 3' end of the linear chromosome and extend that 3' end with repetitive DNA. So this is how that would work. The telomerase enzyme would come and bind to the 3' end, and bringing along with it its own template would then synthesize DNA, 5' to 3', matching this template. Right? So you can see the base pairing rules are the same. So this is DNA being synthesized and added on to the 3' end of the chromosome itself, but using this sequence as a template. Now, because the sequence of the RNA template itself is repetitive, then effectively what happens is the telomerase synthesizes a little stretch of DNA and then shifts over, and the RNA template is going to match some sequence of that because it's repetitive, and that's going to allow the telomerase to add a little bit more, and then it would shift over, and that sequence would add what was just, that sequence would match what was just added, and it would just do that over and over and over again until thousands of copies have been added. And depending on what eukaryotic organism you're looking at, you're going to have different specific sequences. This happens to be the sequence in human, but other organisms will have different specific sequences that get added on. But as you can see, what you end up with is just this repetitive GGTTAG, GGTTAG, GGTTAG added over and over and over again, because that's the complementary sequence to what the template is in the telomerase enzyme. And so we get thousands of copies of this added to the end of the chromosome, and once again, it doesn't make it so that the end replication problem doesn't happen. It just means that we're not losing real genetic sequence with every round of DNA replication. All right, so a couple of things about this telomerase enzyme. When do we see it being active? Well, under normal circumstances, the only time we see telomerase active is early in embryonic development. So in other words, when an early embryo is formed, then telomerase is activated, and in every cell in that early embryo extends the 3' ends of those chromosomes in those embryonic cells. And then telomerase gets shut off under normal circumstances, all right? And so what that means, because telomerase is shut off, is that with every round of DNA replication, those 3' ends are going to get shorter and shorter and shorter. Now, the exception to that is that there are populations of cells that we've referred to before, which are stem cells. And these are undifferentiated populations of cells that divide throughout the life of the organism and are constantly replenishing and replacing particular cell populations in the body. So stem cells are sort of unspecialized in a way, and their whole job is to divide and produce other populations of cells. And so in stem cells, telomerase is active throughout the life of the organism. But other than that, under normal circumstances, telomerase shuts off in early development. And so telomeres, the 3' ends of our linear chromosomes, get shorter and shorter and shorter with every round of DNA replication. Now, it turns out that those telomeres and shortening of telomeres is actually a signal to the cell when it gets to a critically short length that that cell should no longer undergo cell division. And so short telomeres can have one of two outcomes. One, something we've talked about before, apoptosis. So the cell could read, oh, telomeres have gotten really short. I'm running into the danger that I'm actually going to get real genetic sequence starting to disappear here, or alternatively, that, I have short telomeres, therefore my DNA has been replicated numerous times and probably has mutations that have been introduced with every round of replication. Therefore, no more cell division for you. You're likely accumulating potentially dangerous mutations to the organism. Go ahead and go through apoptosis. The other possibility is something called senescence. And senescence at the cellular level means no more cell division, right? So one of these two things is going to occur when telomeres get to a critically short length. There are some researchers who think that senescence at the cellular level, so no more cell division, is actually related to senescence at the organismal level, which is more a reflection of what we refer to as aging. Right? And aging is really just a deviation from homeostasis. Under normal circumstances in youth, the attrition of cells, lost, damaged, old cells that are removed from the body are replaced at an equivalent rate. But what happens with aging is that you get more loss than replacement. So there's a deviation from that homeostasis, and that occurs in all the systems of the body, and that's what we call aging. So this cellular senescence, no more cell division, is related to the senescence that we're seeing at the level of the organism that we associate with aging. And so things like loss of strength, loss of fertility, increasing rates of disease and disruption in the body. But we have to recognize that not all organisms follow the same aging pattern that we see happening in humans and that we're so familiar with. So there are organisms that exhibit something called negligible senescence. That is, they don't appear to age. It's not to say that they're immortal. Obviously, they can be affected by injury, by disease in the same way that any other organism can, but they don't appear to have a finite limit to their adult lifespan. They have negligible senescence. So, for example, lobsters, there are other organisms as well, things like rockfish exhibit this, but these organisms don't exhibit a loss of strength or a loss of reproductive capacity as they get older and older and older. They get bigger, they get stronger, they get more fertile. The males become, even more dominant as they get older. And it turns out that one of the things that's true about organisms that have this sort of negligible senescence is that they have telomerase active throughout their lifespan. And so it's possible that that negligible senescence is associated with the continual reelongation of their telomeres throughout their lifespan. So they don't get this signal of short telomeres leading to apoptosis and senescence, and therefore that inability to effectively replace and repair worn out tissues. So it's an interesting idea. There are a lot of researchers who think that that's not the case, because it turns out that there are sort of other mechanisms that lead to senescence besides telomere shortening. And so there's a lot of debate in the field. I've given you a couple of links in the PowerPoint if you're interested in investigating that a little bit more about telomeres in organisms with negligible senescence. There's also a group of researchers, I should say, that's trying to engineer negligible senescence in organisms that do normally age, like humans. So there's a whole research field that's devoted to trying to overcome the problem of aging and extend human lifespan dramatically by understanding these sorts of molecular and cellular signals that cause normal aging. Okay, so telomeres are a really interesting thing, and they're really important biologically in terms of maintaining the integrity of the genome within the cell. And I just want to end by talking a little bit more about telomeres in general and their structure and their function, because they are critically important in the cell. So when we think about the structure of the ends of our linear chromosomes, they kind of have a structure that looks like this, where they have, the single-stranded bits. So we've got our double-stranded DNA. And then they have this extra bit that's been added on, our telomere, which sort of loops around, and we end up with something that kind of looks like that. So the DNA, so there's the 3' end that's been extended, and here's the other 5' end of the other DNA molecule that I sort of hid there. So we have this sort of looping around of DNA, and then a whole set of proteins that bind to those ends of our chromosomes. And so, somewhat confusingly, both the extended bit of DNA and this whole complex as a whole are both referred to as telomeres. And these capped ends of our chromosomes, you can kind of think of them like the structure at the end of your shoelaces, right? It's a little bit of a cap on the end of the shoelace. And just like that little cap on the end of your shoelace prevents damage and prevents problems from happening with your shoelace, these telomeres are kind of at the molecular level in cells doing a similar thing. So in a cell, if a cell were to find DNA that had uncapped ends, so just DNA floating around in the cell, in the nucleus, let's say, that had a structure that looked like this, no telomeres associated with it, what that would mean to the cell is DNA damage, broken DNA. And what the cell would then try to do is take that broken bit of DNA and attach it somewhere else in the genome so that we don't lose what's, likely to be important genetic information. So broken DNA, naked ends of DNA, is read in the cell as damaged DNA, fragmented DNA, and there's something that's called end joining repair that happens in response to that, where the cell basically says, oh, broken DNA, I don't know where that came from, but I don't want to lose it. I'm going to stick it over here and enzymatically attach it to this other bit of DNA so it doesn't get lost in the next round of cell division, right? So telomeres, by capping the ends of our chromosomes, prevent the cell from inappropriately trying to join chromosomes together in that sort of repair process, all right? So in addition to providing a benefit in the context of the end replication problem, they're also really important for preventing inappropriate end joining repair from going on. All right, and so we could talk forever about telomeres and how interesting and how important they are and how they're dealt with by the cell, but we'll leave it at that.